I'm gonna tell you today about the work that we're doing using stem cell-derived retinal organoids for therapeutic development. As you heard, my lab is part of Cell-Sight, and Cell-Sight is the ocular stem cell and regeneration program in the Anschutz Rogers Eye Center. The mission of this program is to catalyze stem cell-based innovation to treat ocular diseases. And the current focus is on the retina. So I'm gonna give you just a little introduction so that we're all on the same page. The retina is the light-sensing tissue in the eye. It lines the back of the eye, and it's part of a central nervous system. So this is a fundus image of a healthy human retina. And the retina is composed of 7 different kinds of neurons and glia that are organized in a very specific manner that is critical for visual function. And one of the perks of being part of a central nervous system is that the human retina lacks regenerative capacity. What that means is that once these neurons are lost, we don't have any way right now to grow them back. And that leads to irreversible vision loss or blindness. And there's this type of diseases can severely affect the quality of life of an individual, beyond the economic burden. We have 170 million people in the world living with age-related macular degeneration, where you lose the central part of your visual field. And we have 1.5 million people living with retinitis pigmentosa, where you develop night blindness and tunnel vision and eventually complete vision loss. And even though there are some treatments that our ophthalmologists can provide to help with some aspects, for example, of macular degeneration, once these neurons are lost, as I mentioned before, we cannot grow them back. And that's why there's an urgent need for disruptive innovation in this field. We need to develop technologies for and discover drugs for the prevention of neuronal loss before we lose vision. And after we have lost those neurons, we need to develop replacement-based strategies to regenerate that damaged retina. And both of these approaches are being undertaken at Cell-Sight. Our main approach is to use these human stem cell-derived retinal organoids for therapeutic development in both of these areas. And so today I'm going to tell you a little bit about the characteristics of these stem cell-derived retinal organoids. I'm going to tell you about the potential that they have to enhance the drug development pipeline, the current limitations of the system, and also the work that we're doing to develop technologies that will enable the application of these retinal organoids to drug development. But first I have to start with iPS cells. iPS cells are induced pluripotent stem cells. People have been working with embryonic stem cells for 40 years already, and we have learned a lot using this kind of models. But in 2006, the first paper was published describing these induced pluripotent stem cells. This was published by Takahashi and Yamanaka. And it was completely revolutionary in biology and in therapeutic development because it showed that you could take an adult mammalian cell that is fully differentiated and you could treat it in a way that can de-differentiate it to behave like an embryonic stem cell, except that it can come from any individual. And this was so revolutionary that this same group won the Nobel Prize just 6 years after the first publication was available. And this was so revolutionary because of the promise of this kind of technologies. Now we can take cells from any individual and almost from any tissue type. Originally they were acquired from fibroblasts, but now you can even get cells from urine, spin them down and transform them to make these induced pluripotent stem cells or iPS cells. And then, the first thing that happened was that researchers started differentiating these cells into the cell of choice in 2-dimensional cultures. And eventually some people started developing these 3D tissues that resemble aspects of the architecture of the native organ. And that's where the word organoid comes from. And this is very important because these organoids can then be used for the development of therapeutic strategies. They can be used to model disease because we can take cells, for example, from a patient that has a mutation that leads to retinal degeneration in this case. And we can make organoids with cells that have that mutation to try to recapitulate that disease in vitro. That can allow us to do drug screening. We can also use organoids for cell-based therapy. That is one of the avenues that we're exploring. And we can even use them in basic science study to study biological mechanisms. The question that we asked several years ago back in the Cantor Soler lab at Hopkins was, can we use these cells to build the retina in vitro? And there was no publication of this kind at the time, so it seemed like a crazy idea. And a crazy idea because I showed you before that the retina has a very complex organization and composition. And I couldn't imagine how you could recapitulate something like that in vitro. But of course I was wrong. And the answer was yes. We were able to differentiate these human iPS cells in a stepwise manner to generate these three-dimensional human retinas growing in a Petri dish. And the way we do that is we start with iPS cells that are lifted and then are cultured in suspension to form these aggregates called embryo bodies that then we treat in a manner that will promote the development of neuroectodermal derivatives. That is a type of embryonic cell that is going to give rise to the central nervous system, for example. What happens after that is that we plate these aggregates on 2 dimensions again, and eventually they start forming these structures that we can easily recognize under the microscope. And what we do at that point is we manually dissect them, we cut them, and we allow them to float, to grow in suspension again for the rest of the culture period. And when you do that, these structures fold onto themselves and form these optic cup-like structures that really resemble the architecture of a developing retina. And this is grown in Petri dishes because they have to be floating, but we've also been able to grow them in 96-well format, which is important for applications such as drug screening. And finally, these organoids continue to mature to acquire more differentiated characteristics of this type of retinas. So these organoids recapitulate the histological organization and cellular composition of the native retina. And you may have seen this picture before. It's from the Song et al. Paper. And it's just beautiful. You can see the 3 layers of nuclei that form these retinas. And this is growing in a Petri dish without us giving it any inducing factors to make this organization possible. I'll show you here how these organoids, this is 7 to 14 weeks of differentiation, are able to form all the different cell types. We have markers here for ganglion cells, here and here. We have amacrine cells, horizontal cells, photoreceptor precursors. And later on, you form the later born cell types as well. You form a very nicely organized outer nuclear layer that is a layer of the retina that houses the photoreceptor cells, the light sensing cells in the retina. You can see here a neuroblastic layer and Mueller glia. And eventually the photoreceptor cells continue to mature, and you can see rods that are labeled in green here. You can see red-green cones, and you can see blue cones. So all 3 types of human cones are present in these retinal organoids. And not only that, but we can also find bipolar cells. And this is important because bipolar cells are the last neuronal cell type to develop. And they are the ones that are going to connect those photoreceptors to the ganglion cells that are the cells that connect the retina to the brain in vivo. They are the last ones to be born. And we have shown here that that we can even find subtypes of these retinal cell types. And I'm going to take the opportunity along the way to acknowledge the people that have done a lot of this work. But the question then was, are these retinal neurons functional? And we could see that these photoreceptor cells could develop these structures that are called inner and outer segments that are the part of the photoreceptor that contains the opsins that are going to take the photons from the light and transform it into a signal that neurons can understand. And imagine our surprise and excitement when we found out that indeed these photoreceptors that are growing in a petri dish are able to respond to light stimulation. And of course, the big question at that moment was, well, how is that possible, right? And the answer is that the only way you can make, as of right now, the only way that we've been able to make retinas that have this structure in vitro is by allowing these cells to do what they know how to do. So these are cells that behave like an embryonic stem cell. We're pushing them to become neuroectodermal. We select the regions that are going to be retina, and then they go on by themselves. We just help them a little bit along the way. So what I'm showing you here is that each of the steps in human retina development, from the specification of the eye field in the anterior neural tube, that is the part that is going to give rise to the retina eventually, to the evagination of the optic vesicles, to the invagination of those vesicles to form an optic cup, to the differentiation of each of these cells in a specific spatiotemporal manner. All of that happens in the culture conditions in the same way that, or a very similar way that it would happen in the in vivo scenario. So all these processes turned out to be a lot more tissue autonomous than we had ever anticipated. And another important caveat here is that this happens in a species-specific temporal sequence as well, which means that if you take iPS cells from mouse, you're going to get a retina in about 4 weeks, a fairly mature retina. If you take iPS cells from humans, they are going to mimic the timing of human retina development. And that means it's going to be months until you can get photoreceptors that are mature enough to be able to respond to light. The experiments that I showed you before were at 180 days of differentiation. Still, the fact that this recapitulates development in such a faithful manner is quite remarkable. And of course, we're not the only group that has developed these protocols. There are other protocols and other groups that have worked on this. And there's more and more reports coming out now doing RNA-seq analysis that confirm that the developmental timeline of these retinal organoids really mimics the timeline of fetal human retina development. And this is a paper that came out a couple of years ago by an Anschutz group at the NEI, but there are others that confirm this as well. But now I'm going to tell you a little bit about what the model doesn't have, because all this sounds very nice, but there are, of course, limitations. And the first thing that may come to mind is the retinal pigmented epithelium, which we call RPE. The retinal pigmented epithelium is normally located in the back of the eye behind the retina, and it's juxtaposed to the photoreceptor layer. The retinal pigmented epithelium has very important functions for the maintenance and function of those photoreceptor cells. And what happens in the cultures, and I can explain later the mechanics of it, is that during development, the pigmented epithelium is continuous to the neural retina. So it's all one long layer, right? And here we're cutting those structures to let them float in vitro. And so there's no attachment to the neural tube of that RP as it would happen in vivo. And so what happens is that the tissue just falls anteriorly and becomes something that looks like a ball here. So the RPE does differentiate, it does form, but it's just not localized to the area where you would normally see it in vivo. The other thing that the model doesn't have is blood vessels. And this is important for thinking about using these models to try to model certain retinal degenerative diseases. Something like retinopathy of prematurity or wet age-related macular degeneration or diabetic retinopathy is more difficult to mimic in organoid models because they have such strong, of course, vascular component. You could still mimic aspects of that. For example, you can induce stress or damage to certain cell types in the retina and use the organoid model to evaluate neuroprotective drugs. But you have to take into account that you're not going to develop a vasculature. And that makes a lot of sense because the vasculature comes from a different embryonic origin. The other thing that doesn't form in the organoids is an optic stock. And that is also because of the mechanics of the culture system. And the optic stock is the structure that connects the retina to the neural tube during development, and eventually the optic nerve forms, leaves the eye through the optic stock. And the lack of an optic stock here means that when the retinal ganglion cells differentiate, their axons cannot exit the organoid to form that optic nerve. And so they do develop axons, but those axons remain within the cavity of the organoid. And the other thing that we don't have here is microglia. Microglia are the macrophages of the central nervous system. And they have, again, a different embryonic origin. They are yolk sac derivatives. So there's no reason for microglia to develop in this kind of model where we push neuroectodermal fates, right? And it's not clear yet whether these organoids develop astrocytes. I'm gonna guess not, but I may be wrong. So in summary for this part, retinal organoids are a simplified system. They do lack several of the associated structures, and that's something very important to consider when you're trying to model certain diseases. However, they are able to recapitulate all the major events in retina development, and they do that in time and space. They have all the major retinal cell types, and several subtypes have also been evaluated in these organoids. And importantly, they can achieve a level of maturation that allows them to respond to light stimulation. And that's why they have so much potential, both for basic science and for translational applications. And we're gonna get into that now. So when we think about translational applications of these organoids, perhaps the first thing that comes to mind is tissue transplantation. And that is, that is something that we are approaching within CellSide, and is a big component of Dr. Cantosoler's program. And the other thing that you can do with these organoids is, as I mentioned before, use them to model disease, especially diseases that are of a genetic origin, where you can take cells from a patient that has that mutation and produce iPS cells that can be used to generate organoids, but there are other ways to model disease as well. And then you can use those disease models for several purposes, for example, to test gene therapy approaches, to test drug delivery strategies and make sure that the specific drug can access a specific cell type, You can do some toxicology studies to see the effect, the potential toxic effect of certain drugs in a human retinal tissue, which is something that you wouldn't do in vivo, of course. And then you can use them for drug screening. And I'm going to focus a little bit more now on the drug screening approach. Now, can we really use them for drug screening? And so the reason I'm saying that is that when we talk about screening drugs, the first thing that comes to mind is testing thousands of drugs at the same time to try to see which ones have an effect on a particular cell type. And that is not, I showed you the amount of time that it takes to get a retinal organoid that has fairly mature cell types. It's a really long time. There's a lot of manual process involved. And so it's not really a model that is ideally suited for this kind of high throughput screens. But the reason I think it's still important is that the drug development pipeline has been pretty leaky. And what I mean by that is that there are hundreds of drugs that come into market every year. But for certain diseases or in certain cases, the majority of those drugs never make it to sometimes they never make it to clinical trials. And even if they do, they may not make it to the market. And part of the reason for that may be that these drugs are identified in a scenario that doesn't really mimic the physiology that you see in humans. They are usually developed in 2D cell culture models or with biochemical assays, and that's very important. But the scenario there might be different from the scenario that you encounter when you go to a human patient. And then they are tested in animal models, which are absolutely necessary to get a drug that's going to be safe and effective for humans. But part of the problem with that is that those animal models not always recapitulate exactly the pathophysiology that you would see in a human tissue. And that's why we think that the best place for organoid models is in the middle of this pipeline to optimize the pipeline by refining the hits that are encountering in high throughput screening and performing preclinical validations such as efficacy, dose curves, toxicity, delivery mode, things that can be explored at a smaller scale, but that can still make this pipeline a lot more efficient, because then to get a drug to clinical trials takes several years, and sometimes it can cost millions and billions of dollars to get all the way to market. And so if you can make that process more efficient and eliminate drugs that are not going to be effective early on, then I think you have a lot of value. But of course, this is not without its challenges. So the challenges for implementing this kind of approach using these retinal organoids to improve the drug development pipeline, some of them are these. The very long generation time that I was telling you about before, the variable throughput that you get in the generation of these organoids, and also the lack of quantitative technologies for 3D cultures. And that is because most of the in vitro technologies that we have today have been designed for 2-dimensional cultures. And it's very difficult to do, to develop quantitative technologies for 3D that can help in this kind of scenarios, and especially when you're thinking about longitudinal studies. Longitudinal studies are studies where you look at the same organoid over time. And most of the technologies that are currently applied to organoids are terminal, meaning that you collect those organoids, you mush them up, and you look at the RNA or protein level, for example. So how can we do better? We developed a 3-part solution to this problem. The first part involves the the development of improved protocols that can increase the yield of organoid generation, decrease variability, and also shorten the time that it takes to get to mature photoreceptors, fairly mature photoreceptors. The second part of this approach is the development of quantitative technologies that can be applied to these systems. And finally, we can use all that and apply it to a unique triple fluorescent reporter line that has been developed by some of our collaborators. And I'm gonna go through each of these points. First, I'm gonna tell you about our efforts in improving the differentiation and survival of these organoids. So a couple of years ago, there was this paper by an Anschutz group at the NEI that showed that 9-cis retinal supplementation can help accelerate photoreceptor differentiation. So we took this information and we applied it to our model that has been tested and validated and has shown all the different cell types and light response. And what we found when we incorporated this 9-cis retinal treatment is that we could improve organoid yield. By yield, I mean the number of organoids that you're left at the end point of your process. Because one of the things that happen during these cultures is that a proportion of those organoids degenerate over time. And the other problem is that if the organoids are gonna degenerate over time, if they don't degenerate at the same rate in your different conditions, then you're gonna have a really hard time doing any kind of quantitative study, screening drugs in anything that is performed over time. It will really mess up your quantitative power. So what we found with this supplementation is that you increase the yield of organoid generation up to 95% at 130 days of differentiation and about 91% by 150, which is better than anything that we have seen before. And additionally, we see that this kind of treatment increases the length of these inner and outer segments that, as I mentioned, are the part of the photoreceptor that has a machinery for light response. And not only that, but the development of those outer segments happens in this kind of treatment a month before you would start seeing any development of outer segments with our previous protocol. And that is something that you can see here at 120 days in culture, 150, 180. You can see how the length of these outer segments is increased. And these are the people that did the work, of course. Wonderful people in my lab. So how about the development of quantitative technologies? This is the second part of our approach, and it involves a technology that measures fluorescence intensity in a quantitative manner in a 96-well plate format that is amenable for drug development assays. This is something that, I put the date wrong there, but this is something that we developed a couple of years ago. This paper was 2017. And basically, we had to find a system that had very specific requirements to make the mark. First of all, it had to have enough flexibility, sensitivity, and speed to be able to use it in medium to large scale throughput. We also needed a plate reader. We're talking about the machine that reads those multi-well plates that had fluorescence intensity top reading so that we could use V-bottom wells. And this is, I know, very technical, but it's very important if any of you wants to implement this kind of system, because that allows the organoids to self-center in the well so that you don't have to be scanning the whole well to find where the organoid is. And that is important because the key part that makes this applicable to 3D organoids is the capacity to do C-dimensional position, meaning that you can focus in this dimension. And that's important because most plate readers are developed to focus in the bottom of the well. And so you lose a lot of sensitivity and resolution when you have a 3D model where the focus is completely different. And then we needed a machine that would have, that would allow us the flexibility to work with a broad range of fluorophores of different spectral characteristics. And so the system we developed, we termed that 3D ARC for three-dimensional automated reporter quantification. And we showed that this system is sensitive and reproducible and has pretty good quantitative power. So basically what I'm showing here is that we could use a system with fluorophores of different fluorescent spectral characteristics, different subcellular localization, nuclear, membrane, cytoplasmic, and we were able to detect all of them in a very quantitative manner. Now, all these bars that you see here are the detection of the fluorescence in individual organoids. So if you look at the error bars, they're very small, meaning that the technical reproducibility is good, but then you see a lot of variability from organoid to organoid. And the way we went around that was by developing a size normalization strategy using a global fluorophore. And sorry, I'm getting a little technical here, but what this allows us to do is normalize all this data so that allows us to really linearize this curve. And that really improves the quantitative capacity of the technology. And then we did some proof of principle to show the quantitative power of the technology. For example, we did, here we did cell sorting to validate that the fluorescence intensity that we get with our technology corresponds to the number of cells that express a particular fluorophore to prove that it's quantitative. And then we applied it as an example to physiological measurements in these organoids that are growing in vitro. So one of the things we did was to show that you could use a technology like this to measure oxidative damage to the organoids. This assay called the hydroethidium measures reactive oxygen species, and we could track that in a quantitative manner by providing an oxidative insult to these organoids. And of course, the ideal scenario for us, can we use this technology to do longitudinal studies to assess, for example, drug response? And this is one example of that, where we were trying to see if we could track mitochondrial membrane depolarization. That is something that happens when the cells are stressed, and then the mitochondrial membrane can get depolarized, and we can track that with a fluorescent marker called JC1. And what we did here was we provided an insult. We exposed the organoids to a mitochondrial uncoupler, and we saw a sharp decrease in this ratio of fluorescence intensity, showing that the mitochondria had been depolarized. And we maintained that treatment and measured at different time points. And then we removed the treatment and allowed the organoids to recover. And when we did that, the values went back to pretty much indistinguishable from controls. So in summary for this part, the 3D ARC system facilitates these quantitative measurements in 3D tissues. It's very fast, so it meets high throughput screening standards, even though we will probably not use it in high throughput. It has a wide versatility of applications and fluorophore selection. We developed a way to account for size variability in these organoids, and we showed that we could perform longitudinal studies. And all that can be done in a human retina context. And so the third part of this approach was to combine both the improved protocol and the quantitative technology to a potential application. And in this case, we chose a triple fluorescent reporter HIPSC line. This line was generated by our colleagues at Miami University. And what this line has is 3 different fluorescent reporters that are expressed in certain cell types in the retina. So you'll have blue expression in progenitor cells and bipolar cells. You'll have red fluorescent expression in photoreceptor cells and green fluorescent expression in ganglion cells. And so we took this line and we generated retinal organoids from that. And we could see that the expression of these different fluorophores really matches the localization of all these different cell types within the retina. And these pictures are in 3D organoids. These are not sections. So we're looking at the whole volume of an organoid here. And I just had to show you this picture because it's pretty, but you can see how these photoreceptor inner and outer segments are labeled with our red fluorescent marker that labels photoreceptors. And this is just one example of how that can be applied. And this is preliminary data that we have, right? But we applied this technology to the quantitative live tracking of these fluorescent reporters during the development of these organoids. And what that allowed us to do was find features of retina development in humans that had not been described before. For example, we could see this is the expression of recoverin, that is a marker of photoreceptor cells, and we could see a steady increase in recoverin expression, as you would expect as the organoids develop and they start generating more and more photoreceptor cells. But what we found, for example, for retinal ganglion cells was that they would start being expressed and then there would be this dip at 8 weeks of differentiation. And then they will continue to increase in numbers. And we were intrigued by that because we knew that there's 2 waves of ganglion cell death that happen during development. And this was happening at the same time that you, or at the same timing during development that you would expect based on our knowledge from other species. But this is happening in vitro. And so we had to confirm this, and this was the work of a grad student in my lab that graduated last year, Tara Brooks. And so she did a lot of studies, and I'm just showing you part of this, showing by a different technique and a different ganglion cell marker, and these are sections instead of whole organoids. She did all this quantification, and she confirmed that she was still finding this decrease of retinal ganglion cells at 8 weeks. And then she did more studies to show that there's a significant peak in ganglion cell death at that time. We've repeated that in multiple batches of organoids, and we've repeated that in different cell lines, and we've found exactly the same. So this gives you an example of how we can understand even mechanisms of development by using this kind of technologies. And we can do all that quantitatively. Other applications that you could think about a program or an assay like that would be, for example, if you were to test neuroprotective drugs. So you could generate organoids that express all these reporters. You could induce cell death in particular cell types. You could induce cell death in a manner that is similar to that you see in disease, for example, by increasing oxidative damage. And then you can track the fluorescence expression over time so that you can see how those cells are dying and how your treatment may prevent that cell loss. And that's a general idea behind this. So, and that's the diagram that I'm showing here, where you have the fluorescent reporter line. You generate organoids with the improved protocol. You could potentially use a particle sorter if you were doing this in larger scale. And then you could use the microplate reader approach to quantitatively measure this expression. You can then do your drug treatment, you can do your disease model, and then repeat and measure longitudinally what is happening with these organoids over time. And this was the topic of a challenge that the National Eye Institute developed a few years ago. And the idea of this challenge was to try to get researchers to think about ways of developing physiologically competent stem cell-derived retinal organoids, and to develop technologies that would enable the application of those organoids to the development of therapeutic drugs. And that's why I'm happy to say that our team won this award very recently. And when I say team, I mean, it's not just my lab. Val Canto-Soler's lab participated on this, and also Mike Robinson and Katia Del Rio Sonis at Miami University and Nanoscope Technologies. And this is just to show off our beautiful facility at Cell Site, because it's always nice to see. And this is the most important slide because all this work was made possible by all these people. So I want to thank all the people in my lab, but also all the faculty, trainees, and staff in our Department of Ophthalmology, because they are the people that make the impossible possible. And all of our collaborators and the funding sources, because we could not do any of this work without that. And with that, I'll be happy to take your questions.